Okay, so good evening everyone. This is your 11th week session, and in this session we'll look into the personalized therapy, in which we'll see the history, that how it began and what are the basics of personalized therapy. Then we'll see that how the genome editing has helped in personalized therapy. So after this we'll practice MCQs, and while discussing these topics we'll also look into the current state of literature, and then I'll also take your doubts. Okay, so first we'll talk about what is personalized therapy. So when we talk about the traditional medicines, which were the previous state of art medicines, so at that time they assumed that same drug or dose can work for everyone. But in reality, we have seen that different patients behave differently to different drugs, and different drug doses can affect persons differently. So Hippocrates in 400 BC, he highlighted the importance of patient's individuality. He mentioned that different individuals have different tendencies to behave towards different drugs, and different doses might be required for each individual. So know what sort of person has a disease. That was the earliest idea of personalization, and it was the foundation for modern personalized therapy approaches, and it moves away from the universal treatment towards patient specific care. So previously people were focusing on a universal treatment, that one drug can work efficiently on each individual, but now we have seen that different patients respond differently, so now we have moved towards the patient specific care. So this is the comparison between the current medicine and the future medicine. So these are the cancer patients who are suffering from the colon cancer, and in this it has been observed that the therapy was given to all of them. So in these patients can be divided into three groups. So in one group, the effect could be means visibly seen, that this drug was showing some effect. Either it is curing the patients, or means some kind of effect could be observed. Then comes the second group, in which there is no effect of this drug. Then comes the third group, in which there is adverse effects. So these patients given the same therapy, but they have shown that they are not curing the cancer, but they are showing adverse effects, so their cancer was worsening. So now what is the future medicine, which is a more personalized diagnostics, in which what we do is we take all these patients and we collect their blood, DNA, urine and tissue analysis. We perform all these analysis, and then we find out that what amount of dosage should be given and what kind of drug should be given to them. So in that way we give different personalized therapy to different group of people, and ultimately we can see the effect on each individual of that particular drug. So this is the how we have moved from one treatment towards the personalized treatment for everyone. So in order to define that what personalized therapy means, so it means the treatment which is designed for an individual, and the factors which are considered while designing this therapy is genetic makeup. So first of all we see that what is the genetic makeup of that human being, what are the genes, and means either there are some mutations or what is the genetic characteristic of that individual. So in this we look at the genotype of an individual. Then comes environment. So we have also seen that the people who are present in different environments, for example the people who are in the desert conditions or hot climate conditions, so they may behave differently to a drug as compared to the person who is living at the means polar regions, or the regions which are extremely cold, which has extremely cold climate. So in that way we also look at the environment. Then we look at the lifestyle, that whether that person is sedentary or whether that person is very active. So in that way that can also affect the drug, because we know that in the later sessions we looked at how the drug reacts inside our body. So that reactions, that all the enzymes and metabolic processes which are taking place, so that also depends on the lifestyle, that what kind of food you take, where you live, what is your means day to day schedule. So everything depends, means that will determine that whether the drug will how the drug will act on that particular person. Then comes phenotype. So these are the visible traits that you can see. So these are also the important factors which must be considered before designing the personalized medicine. So the aim of this personalized therapy is to increase the efficacy while minimizing the toxicity, and in this case we tailor the drug, and we also decide what amount of dosage should be given. It prevents unnecessary side effects, and it also forms the basis of the precision medicine. So many times the personalized therapy and precision medicine, they are used interchangeably. So personalized therapy is a broader term, and precision medicine is like the medicine which can behave precisely on that particular individual is the precision medicine, and personalized means that is specific to one particular individual, that is known as personalized medicine. So this is how the personalized therapy works. So same thing is shown here, that different individuals, as you can see here, the phenotype, their genetic makeup can determine what amount of dosage is required to them. So some can means the drug can show effect. So in this case, this particular lady is given just the one pill of the drug, so one capsule is sufficient for her, whereas this individual, he requires just the half of that particular drug, whereas this individual who was handicapped, so he required two capsules or two doses of this particular drug, whereas this lady, she does not require that particular drug because this drug might not work on her. So this shows that how these studies can help you to determine that what amount of dose could be sufficient for that particular individual. So now the question arises that why the personalized medicine is needed. So drugs often fail in large subset of patients, and that is also one of the main reason that we have seen that till now we do not have a perfect cure for the cancers, because different individuals behave differently. So on one particular subset of patients, that drug might work well, but when you give it to other subset of patients, it might not work at all, or it might have some adverse effects. So in that way, that was observed by different scientists. So later on they understood that we cannot give universal medicine to all the people. So they realized that, for example, 25 to 60% patients respond effectively to common therapies, and then adverse drug reactions were also observed, which lead to the morbidity or mortality. So many deaths were also observed. So later on it was realized that how important is this personalized medicine is. So trial and error prescribing is inefficient and unsafe. Personalized medicine reduces the cost of failed therapies, and it also improves the patient satisfaction and long term health outcomes. So these are some of the applications of personalized medicine. So in this it is shown that how the personalized therapy works. So for example, we have two patients, patient A and patient B. Now their genes can be different. So let's assume they are suffering from some cancer, and there is a gene which is responsible for that particular cancer trait. So there could be different mutations in that particular gene. So in this patient there is mutation here, whereas in the other patient there is mutation B, which is present on the same gene but at some other place. So now drug A can work well if the mutation A is happening, whereas drug B can act well if mutation B is there. So in this case, if we know what kind of mutation the patient has in his gene, then we can give a proper drug to that particular patient. So this is just one single description that how the genetic makeup can determine what kind of drug can be given. So this is just one highlight of the personalized medicine therapy. Similarly, in this case also, we are comparing between the conventional medicine and the personalized medicine. So in conventional medicine, the same medicine is given to all the patients, and then it is observed that some patients shows the serious adverse events, whereas some do not show any effect or very low effect of that particular drug, whereas other group of people shows good efficacy towards that drug. Whereas in case of personalized medicine, we design it in such a way that all different groups of patients or subset of patients, the adverse effects are very low. So if we can see in this graph, it's very limited. No and low efficacy is also limited as compared to the conventional medicine, whereas good efficacy uh, bars are quite high. So this is just to show you that the outcome of therapy has improved if we have shifted from the conventional medicine towards the personalized medicine. Okay, now we'll talk about the genetic variability in drug response. So because now we are talking about the personalized therapy, that what are the different factors which are considered while designing the personalized therapy. So one main factor which we have seen is the genetic makeup, that how the genes can determine what kind of drug can act well on that particular patient. So in this we'll see that how this genetic variability respond towards the drug. So DNA variants, that means if a gene has different variants, or there are some SNPs which are the single nucleotide polymorphism, which is normally present, which is not related to any disease, but that is just a polymorphism of that particular gene. So DNA variants can lead to differences in the metabolism, absorption, excretion, And single nucleotide polymorphisms are the most common variations. So whenever we go for the DNA fingerprinting or all those things, so we know that majority of our DNA is entirely same, but some of the regions vary. So how do they vary? So they vary due to these SNPs or other repeats and the number of copies of those repeats which are present. So these are the main things which leads to the variations among the genetic material. So for example, there is a gene which is cytochrome P450 enzyme family which metabolizes 75% of the drugs. So now this is the enzyme, and enzymes are like the proteins only, so they are the proteins. So how these proteins arise? They arise from the gene. So there might be some gene which is giving rise to the, means which is a DNA which give rise to RNA, and then this protein is formed. So in that way, if there is some problem at that gene, of if there is some diversity at that level of that gene, then the metabolism will also vary among different individuals. So in that way, they are mainly focusing on this particular gene because they are saying that it is responsible for the metabolism of 75% of drugs. So if we compare this particular gene among different individuals, then we can get to know that what is the metabolic rate of that particular individual. So what amount of dosage should be given to that particular patient. So then comes poor versus ultra rapid metabolizers. So in that way, if a patient who is having a poor metabolism, so in that case we need to decide how much drug dosage should be given to him. So there are chances that it means it becomes underdosing, that we are not giving enough dosage, or it could also lead to the overdosing risks. So that's why it's very important to study this particular gene family and see that what is the nature of that particular person, whether he is a poor metabolizer or he is a ultra rapid metabolizer. Genetic testing helps to identify at risk patients, and it also forms the core of pharmacogenomic studies. So this also means the same. Pharmaco means when we are talking about some drugs, how they are behaving, and genomics means how these drugs are linked to the genetic makeup of the patient. So in this case, let's talk about the patient and their SNPs. So in this, different individuals are compared and their SNPs are compared, that this particular region where the nucleotide varies. So this is the allele, this is the enzyme. So enzyme is same, but due to this particular change, due to this particular one single nucleotide variation among all the four individuals, their metabolism is very different. So this particular individual with adenine, he is having ultra rapid metabolism, whereas this lady with C is having extensive metabolism. The person with the G nucleotide is having intermediate metabolism, whereas the lady with the T, which is thymine, so she is having the poor metabolism. So from here you can get the idea that these individuals having just one single nucleotide variations and they have different metabolisms. So that means they will behave differently to that particular drug. They will metabolize it at their own pace. So in these conditions, it's very important to see that what is the metabolism, so that we can design the dosage of the drug, so that it does not become toxic, or it should not be underdose, that it does not behave or it does not show any effect. So the patients with the same diagnosis, the same prescription, and without pharmacogenomic stratification, so if we do not do this particular classification, if we don't do anything and we give the same dosage to each individual, so then we will see that there are different groups of individuals. Some will show good response with severe adverse effects, whereas there will be one group which will show good response with severe minimal effects. Then there will be one group which will show the null response but without adverse effects, and there could be another group which shows null response but with adverse side effects. So these groups can be seen if we do not focus on their metabolism or other characteristics, and we are blind and we are giving same diagnosis and same medication to all the individuals. So this is just to show you that how the genetic variability is playing an important role in the drug response or drug effect. So now we'll talk about the historical roots, that how this medical field advanced through years. So if we talk about historical roots, then in India, Ayurveda was the most primitive science of medicine. So it is about 5,000 year old, and it emphasized on the prakriti, which is the individual constitution. So means right now we are talking about the personalized therapy, but this is not a very novel term or very new thing. Previously also, when the Ayurveda was there, so that also focused on the individual constitution and individual personalized treatment. So now this Ayurveda is based on the prakriti. it compares the body type of a particular patient, and that way it design the drugs. So in this case, in this Ayurveda, we have three prakritis. One is Vata, another is Pitta, and third one is Kaph. And then there are some individuals which are like combination types, which are like Vat, Kaph, or which have, these are known as the three doshas also. Then some has Vat, Pit, which is the combination of Vata and Pitta, and then Pit, Kaph, which is the combination among Pit and Kaph. So there is a three dosha therapy, and it says three dosha theory, which says that there should be balance between these three doshas, that is Vat, Pitta, and Kaph, and treatment is tailored to patient's dosha imbalance. So in Ayurveda, how they treat the patient is they firstly study their these three prakrutis, and then they see that whether it is balanced and where it is, what factor is leading to the imbalance among these three, and how, means which drug can bring back that balance. So that is how the treatment was designed previously. So early recognition that individual differences impact therapy, and it is considered one of the oldest systems of personalized medicine. So this also shows that how our primitive, or means ancestral culture is so much rich in this modern information, which we call now modern, but it is no longer modern. It was already mentioned in the scriptures or literature which was previously present in our Hindu mythology. So when we say Pit dosha, so it is the combination of fire and water. And then here are some of the qualities which are mentioned. So the Pit dosh qualities are, they are slightly oily, penetrating, sharp, hot, light, and flowing. And their skin type is oily, and it is warm to touch. Their hair are medium, full, may bald later. Body frame is medium and stocky. Their bowel movement is regular and it tends towards loose. So these are some of the characteristics that how one can identify what kind of dosha is more prominent in that particular individual. So each individual is different. Then in Kapha, we have the combination of earth and water, and the qualities are cold, heavy, sticky, stable, moist, dull, smooth, soft, and these are some of the body characteristics. And then in Vat dosha, this is the combination of air and ether, and the qualities are light, cold, rough, dry, mobile, and subtle. So, if you're interested, you can go through these body types. But what I want to tell you from this slide is that these three doshas, that means these are the three different characteristics. So these tells the characteristics of a person. So in that way, each individual has different characteristics, and similarly different drugs will be given to them. Different drugs or different medications can be given to them based on their body type. So this is one such type of personalized therapy only. So Ayurvedic pharmacology, which is also known as Rasa Panchaka. So there are 5 principles, which is Ras, Gun, Virya, Vipak, and Prabhav. So Rasa means taste, Gun means quality, Virya means potency, Vipaka means digestive effect, and Prabhav means specific action. So right now the pharmacology which we study for each individual, that what is the pharmacology, different pharmacological studies of drugs comes up. So what they ultimately do, so they check all the properties. So similar principles are already mentioned in Ayurveda, in which they've already seen that what is the taste of that particular medicine, what is the quality, how effective it is, what is the potency. So potency is also the potential of that particular drug to perform its action. Post digestive effect, so means after digestion what happens, where it moves, means to which tissues it moves, and what happens, either it is excreted. So this all comes under pharmacological studies only. So this is the Ayurvedic version of pharmacology. And Prabhav means specific action. So personalized selection of medicines are based on these factors. So we compare these things and then we design the personalized selection for the medicine compared to modern pharmacological concepts. So modern pharmacology is also having the similar concepts. It also believe on dose, what is the mechanism of that drug, and whether it will lead to some side effects or not. And then these are also the properties of the drug only. So there are admit properties which are not discussed right now, but these properties are the drug properties which must be seen before designing the drug. So there are different small molecules or chemicals which are already available, and then we check that whether it can behave as a drug or not. So the properties are like ADMET properties, in which we check for the absorption of that drug, desorption of that drug, metabolism of that drug, excretion, toxicology. So there are five main characteristics which are seen, and then there are different other properties also. So there is Lipinski rule of 5, in which we check that what is the molecular weight, what is the polarity of that drug. So all these factors come under pharmacology. So before calling a small molecule or chemical as a drug, we check these properties, that either it is a means it will work properly or effectively inside the body, or whether it will lead to some toxic production of chemicals. So same thing is mentioned here, that if we compare the Ayurvedic pharmacology with the modern pharmacology, then the concepts, basic principles are the same, and it shows continuity of personalized medicine across centuries. So this shows that this is not a modern concept, but it already existed and it is still going on. So this is the current medicine system, in which group of patients having the same disease, modern diagnostic methods are available, same therapy is given for all. So as we discussed, that there could be three different groups, which we will see that some are benefited from that drug, some do not show any response, so there is no benefit, whereas other people show adverse effects. So concepts of personalized medicine in Ayurveda, so in which there is division of group of patients based on their prakriti in the same disease. So for example, if a patient is suffering from cold or fever, so we'll see what is the body type of that particular person, we'll see his prakriti, that whether he is coming from the kapha type, with type. uh, so in that way, vat, pit, kaph, all these characteristics are studied for that particular person. So on that way we divide the patients into these three groups, three prakritis. And there is Ayurvedic diagnostics, so in which there are Ashtavidh Pariksha, the eightfold diagnostic methods. So these are also the eightfold methods which exist in Ayurvedic concept, in which they check for these eight properties in that particular person. So this is a holistic therapy according to different constitutions. So medicine, biopurification, diet, and lifestyle regimens. So all these things are given as a therapy. Medicines could be given, some diet or lifestyle changes could be asked that particular person to follow. So each patient benefits from the individualized treatment. So this is the comparison between the Ayurveda and the personalized medicine. So both are having the similar characteristics, both are same. So global ancient medicine systems. So now we'll talk about that, what about the other countries, or the other countries in the globe. So if we talk about Egypt, there is a book named Eber's Papyrus in 1500 BC. So it has the detailed prescriptions and disease remedies. So this is the excerpt of that particular book. This is how it looks. So this is the, means in this we are talking about what were the ancient medicine systems in other countries. So it is about the Egypt, which is about 1500 BC. Then in Greece, Hippocrates was a scientist who mentioned that disease is caused by the imbalance of humors. So as in Ayurveda we talk about these three doshas, so similarly Greece, the Hippocrates also talked about the four humors, but the humors are like these yellow bile, black bile, blood, and phlegm. So he said that the body of a person is made up of these four humors, these four liquids. So these liquids are yellow bile, black bile, blood, and phlegm. So these are the constituents present in each individual, and they are balanced. If their balance is disturbed, then a person shows disease. So this was the theory which existed in the primitive Greece. Then comes India, in which Ayurveda we talk about the constitutional medicine, which is the dosha system, and we have already seen three doshas. And then Indian medicine system is also divided into five main types, which is Ayurveda, homeopathy, yoga and naturopathy, Unani, Siddha. So they come under Ayush. And in this case, the most primitive one is the Ayurveda. So that's why we are talking about Ayurveda only. Then comes China, in which the traditional Chinese medicine was based on yin yang and ki balance. So they also believed on these properties, that in each individual there is a yin and yang factor, and in each yang there is a small yin which is present, and they believe that we can change the, if we balance those states, then we can treat the individual. So these all are somewhat type of the individual treatments only, in which each individual has different constitution of each components. For example, in this case, each individual has different combination of humors. So one way or the another, they also talked about the individual variability, that all these systems, they emphasized on the individual variability in diagnosis and treatment, and early examples of integrative and personalized approaches. So to some extent, these all points towards the personalized treatment, that there was some ideology which primitively existed about these personalized treatments. Now we'll talk about the Garrett's contribution, Archibald Garrett's contribution towards this particular treatment. So in 1902, he talked about the inborn errors of metabolism, in which he studied alkaptonuria. So in this particular disease, the color of the urine turns black, and this is linked to the metabolic defect. And it is the first connection between the genetics and disease. So if we look at the gene, and if we see that there is some change in that particular gene leads to this particular disease state, so he mentioned the combination or connection between the genetics and the disease. He pioneered the concept that inherited factors affect the drug metabolism, and he opened the path towards the modern pharmacogenetics, in which means when we design the drug, we just not only look at the drug, but we also look at the genetics of that particular person, and that is known as pharmacogenetics. And it marked the shift from the descriptive medicine to molecular understanding. So in this particular alkaptonuria, how it happens, so in our body, the phenylalanine converts to tyrosine, and which further converts to 4-hydroxy phenyl pyruvic acid, and then this converts to homogenetic acid. If this particular homogenetic acid does not convert to melyl acetoacetic acid, if it does not convert, then it accumulates in the body and it goes to kidneys, from where it means adds the black color to the urine. So that is how the alkaptonuria happens. So the enzyme which is responsible for this conversion is defective, and it is been shown that there is a mutation in the gene, and this particular mutation is inherited in the autosomal recessive manner, and the enzyme which is responsible for this conversion is homogenetic acid oxidase, which oxidize this homogenetic acid to melloacetic acetoacetic acid. So if this particular enzyme or this particular gene is defective, then this conversion does not take place, which ultimately leads to the alkaptonuria. So this is the metabolic disorder. There is defect in the metabolic gene which is responsible for this conversion. So this is how he mentioned that means all these gene genetic studies are also important while designing the drugs, and this also determines how the patient will behave to the drug dose. So this is the timeline of medicinal or medical history, that how it began. So there was a prehistoric medicine which was based on the herbs, rituals, trials and errors. So primitively, when there was no proper well documented medical system was present, so people, what they used to do whenever they used to suffer from any disease, they used to take some herbs, and then they used to see whether it will bring about some positive effect or not. So on the basis of trial and error, they used to take the means drugs, we cannot call them drugs, but they used to take those herbs in response to different disease states. Then come ancient systems in which Ayurveda, Chinese medicine, Egyptian papyri and Greek humoral theory comes in, which we have already discussed. Then in 19th century, there was a scientific revolution in which different scientists were doing different research. So then germ theory came, microscopy was also discovered, and anesthesia concept was also discovered, that anesthesia can, if it is given to a person, then we can do different other operative tasks, or some operations can be done without causing the pain, because many people that means during the operations the pain was so unbearable that people used to die due to pain. So anesthesia was a, when it was discovered that different herbs can be used as anesthetic, so it was realized that in this case we can treat a person without causing the pain so that they can survive. So this is just to repress the pain. In early 20th century, discovery of vitamins, hormones and insulin. In 1953, Watson and Crick gave the DNA double helical model. In 1970s to 1980s, recombinant DNA technology came in, sequencing methods came in. So this is showing that how the advancement took place. So from the herbs, how we moved into the modern chemicals, vitamins, hormones, and then from there we also moved into the personalized treatment where we look into the individual DNA, and with the recombinant DNA technology, we could also means recombine that DNA. We can correct that DNA, and we have also seen the gene editing tools. So they come under recombinant DNA technology only, in which we are working with the DNA, we are recombining it. And then sequencing methods came in 2000s. Human Genome Project was also almost completed in 2003. So that was the milestone for personalized medicine, because now we know that which genes are present in human and which gene are responsible for which disease. So today the integration of genomics, AI and biotechnology is shaping the precision medicine. And this is also the similar timeline which is shown, which we have discussed. So there was a case study of malaria and pemaquine. So pemaquine is a drug which was given to the malarial person. So it is a early anti-malarial drug. In 1920s it was identified. So it was found effective in many patients, but it caused severe hemolysis in some. So this is a case study. In this we are looking that why do we need the personalized treatment, why we need to study the individual before giving any drug. So in this they observed that some patients, they were showing better effects, it was effective, it was anti-malarial effect, in some patients it showed, but in other patients it led to the severe hemolysis. So later it was discovered that the patients with the G6PD deficiency at risk, so the patients who were having this particular deficiency, they were at the risk of this particular drug. So in those patients, if we give pemaquine, then it leads to hemolysis, that is breakdown of the blood cells. So it is the first demonstration of drug response linked to a genetic variation. So that means that drugs cannot be given without looking at the genetic makeup of the patient. And it highlighted that there is a need for the genetic screening before prescribing any drugs. And it established the concept of personalized therapy in pharmacology, and it prevented unnecessary deaths once linked to the enzyme deficiency. And today, G6PD testing is routine in malaria endemic regions. So, in whatever region, if there are many cases of malaria, so before prescribing any drug, we first look into this G6PD testing. This particular gene is tested, that whether it is defective or whether it is working properly. Okay, so this is the structure of this particular pemaquine drug. So drugs are generally the small molecules. These are the chemical compounds. So in this case, it has a heme binding group, and these are some of the functional groups which are responsible for reacting with our proteins in our body. So okay, so if you're interested, you can go through the structure. So now what happens? So this shows that the malarial parasite, it prefers the normal RBCs and it attacks the normal RBCs. So if we have this drug pemaquine, so pemaquine comes under the quinolines. Quinolines is the bigger family which contain this particular group. So now in this case, we will replace this term with the pemaquine. So if we have a pemaquine drug, so it leads to the oxidative stress, and it leads to the anti-malarial action in that particular cell, that particular RBC where the malarial parasite has attacked. And similarly, this particular drug, because it is causing the oxidative stress, so it could also lead to the hemolysis. So if G6PD is deficient RBC, so if the RBC is deficient in this particular gene, so it will lead to the hemolysis of that particular RBC. So in this case, this particular deficiency, the patients with this particular deficiency will, this particular drug not only affect the RBCs which are suffering from this malarial parasite, but it will also lead to the hemolysis of all those RBCs which are deficient in the G6PD gene. So in the patients which has this deficiency, they will have all those RBCs which are deficient, so it will also lead to the hemolysis because it will increase the oxidative stress, which these normal, because the normal RBC does not exist, G6PD is missing, so they are not able to fight against this oxidative stress and they will ultimately break down, they will lead to hemolysis. Okay, so this is the another article which is obtained from this particular paper. So if you're interested, you can go through this, in which they have in detail explained that what is the mechanism, how this deficiency is linked to the hemolysis. So in normal scenario, in the glycolysis, as we know, that the glucose is converted into glucose 6 phosphate, and this glucose 6 phosphate later on with the help of glucose 6 phosphate dehydrogenase, it converts to D-glucono-1,5-lactone 6 phosphate, and then it enters into pentose phosphate cycle. And in this case, this conversion requires the NADP positive converts to NADPH. So there is a reduction of NADP positive to NADPH, and it is a reversible process, so it again converts back. So in the case, it is showing the antioxidant defense. It leads to cell growth and redox regulation of proteins. So this is the case where the glucose 6 phosphate is present. So it provides antioxidant defense. It leads to cell growth and redox regulation of patients. So now assume if we do not have this particular gene, then it leads to the, if we have this deficiency of glucose 6 phosphate dehydrogenase, then it means that we will not be able to convert glucose 6 phosphate to this D-glucono-1,5-lactone 6 phosphate. So we will not be converting the NADP positive to NADPH, and now these particular other proteins which are associated, this conversion will also not take place. So in that way, it will not have any antioxidant defense. So now if that particular drug will increase the oxidative stress in the cell, so our cell is not able to deal with that particular stress, due to which the hydrogen peroxide will increase in our body. So in this case, hydrogen peroxide which was formed, it is converted to water, and then oxygen is separately released. So but in this case, because these reactions are not taking place, so we will not be able to convert hydrogen peroxide into water. So hydrogen peroxide will increase inside that particular cell where we have given the drug, so which will lead to the hemolysis, breakdown of that particular RBC. So this is at the level of, means this is at the chemical level, that how the different reactions are responsible for the hemolysis if they do not take place then. So this is the G6PD deficient PRBC. In this case, if we give the drug, then the first of all, even without the drug, the H2O2 will increase, because normally also oxidative stress can be present in the cell. So the drug will just increase that oxidative stress. So it is this particular chemical is released, which is ferriprotoporphyrin 9, which converts to hemozoin and which is toxic to macrophages. And then hemichrome deposits on the PRBC membranes, which increase the phagocytic removal of PRBCs in vivo, and this also increase the oxidative stress in parasites as well as host cells. So increased oxidative stress in parasite and host cell, which will also kill the host cells. So in this way, although it will kill the cells which are affected with the parasite, but it will also kill the normal cells. So that is the disadvantage of giving that particular drug to the patients who are having the G6PD deficiency, which is glucose 6 phosphate dehydrogenase. And if you're interested, you can go through this, in which they have talked about the this particular inheritance pattern of this gene, that the males are the hemizygous and the females have the 2 copies of this gene. So in that way, females can still act as the carriers, but if males having the defective copy of that gene, then they will always be the patients, they will always suffer from this deficiency. And this shows the different countries and what is the rate of this particular deficient G6PD alleles worldwide. Okay, so India is also there. Okay, so till now we have looked into the means different concept, that why the genetic variations are important before giving any drug. So now we'll talk about the pharmacogenomics, because we were talking about the drug effect and how genetic variations can vary the effect of each drug. So in pharmacogenomics, we study that how the genes affect the individual drug responses. So when we say the pharmacogenomics, that means we are talking about the entire genome. So it focuses on the entire genome versus single gene approach. So there is another term which is known as pharmacogenetics. So when we say genetics, we focus on the single individual gene. When we say genomics, we talk about the entire genome. So it enables the design of personalized drug regimen, and it reduces the trial and error prescribing. It also saves the cost and time, and it supports the safer prescribing, and it avoids toxicity, because in the primitive case, as in the previous case, as we have seen, that if there is the deficiency in that particular gene, then how it can be toxic if it is given to those patients. So if we know this particular genetic makeup of that person, then we can avoid the toxic effect of the drugs. So it integrates genomics, bioinformatics, and clinical data, and it plays the role in cancer therapy, psychiatry, cardiology, and infectious diseases. It is the core component of modern precision medicine. So this is the genomics, and pharmacology means where we study the medicines. So if we combine both these fields, it becomes pharmacogenomics, in which we see that how a client's genetic variations affect their response to medications. So it provides individualized and safe care. Similar thing is mentioned that this person responds to the normal dose, whereas this person he responds to the lower dose based on the genetic makeup, whereas this person requires the higher dose, whereas it requires this particular lady requires the alternative medicine. This medicine might not work on her. So this shows that how the genome of each individual can be different, and how they can behave differently to the different drugs, and to the same drug they behave differently. Okay, now we'll talk about pharmacogenetics versus pharmacogenomics. So pharmacogenetics is single gene study of drug response, and this is a older term. Pharmacogenomics is the genome wide approach, and it is a modern term. For example, in the pharmacogenetics we look at the cytochrome P450, and in this case cytochrome P2D6 variants affect the codeine metabolism. So as it was mentioned that this particular gene family is responsible for the drug metabolism. So previously the studies were only on this gene and not on other genes, but later on it was realized that not just one single gene but other genes could also affect the influence the metabolism of that particular drug. So later on the term moved towards the pharmacogenomics, so which is a genome wide studies predicting the chemotherapy toxicity. So chemotherapy is usually given to the cancer patients, and not every cancer patients shows positive effects towards that chemotherapy. So with positive effects I mean that the reduction in the cancer tissues. So in many patients, the even after the chemotherapy the cancer status again reappeared. So, and chemotherapy is also toxic in some patients. So if we look at the entire genome of that particular person, then we can reduce this toxicity, and we can also predict that on which patients it can be toxic. So this shows that, but both these pharmacogenomics and pharmacogenetics both aim to improve the drug safety and efficacy. Pharmacogenomics allows the use of high throughput sequencing and artificial intelligence, whereas in personalized medicine, it now uses the pharmacogenomics as a standard term. Pharmacogenetics is like the roots, whereas pharmacogenomics is a broad field. Okay, so this is again another comparison that how we are progressing from pharmacogenetics towards pharmacogenomics and then pharmacoomics. So not just genes but transcriptomics, proteomics, metabolomics. So all other omics are also studied in this pharmacoomics, which is the most modern term. So similar thing is mentioned in all of them. In this case, it is shown that the pharmacogenomics can predict the drug dose. In that way, if you predict the proper drug dose, then it can prevent the adverse drug reactions, and it can also improve the efficacy of that particular drug. It can pharmacogenomics can help to develop the targeted drugs for cancer therapy. It can enable the drug development and discovery. It can predict the activation of prodrugs. So these are some of the applications of pharmacogenomics. Okay, now we are similarly talking about the genetic variations and drug response. So variation in metabolic enzymes, for example cytochrome P450 family, is very crucial. So the patients who are poor metabolizers, so they require, if higher drug levels are given, it can lead to toxicity. Whereas if the person is ultra rapid metabolizer, then drug is cleared too fast. So that means if the low dose of that drug is given, then it is ineffective to that particular person. So variations affect the absorption, distribution, metabolism, and excretion. So these are the ADME properties. So there are total 5 properties. In this only 4 properties are discussed, which are absorption, distribution, metabolism, and excretion. So if there is variation at the gene, because if the person have different metabolic characteristics, then the absorption of the drug will be different. The how that drug get distributed to different tissues throughout the body will also be different. So metabolism is also one important factor which will decide that how this drug is broken down inside the body. And then comes excretion, that after its absorption, the whatever the leftover is, how well it is excreted out. So the variations affect all these properties, and it also impact the drug targets. And drug targets means these chemical compounds, because they are reactive in nature, so they go to some site in the body and they react with the proteins. So proteins where the drug binds or where the drug reacts are known as the receptors, which could either be enzymes or proteins. So it explains that why the standard dose doesn't fit everyone. Okay, so now this shows you, because each individual has different metabolism, so standard dose will not effectively act for all the individuals. So testing patients before prescribing is very important to design the safe therapy, and it is the core principle behind the precision dosing. So this is the determined genotype. So this is the comparison among the genotypes for this particular gene. So predicted phenotype, after looking at this genotype, individuals can have different phenotypes. Some could have ultra rapid metabolizers, some could be extensive metabolizers, some could be intermediate, and some could be poor metabolizers. So ultra rapid means they are too rapid in drug metabolism. So if particular dose is given, so it might not show any response. Whereas extensive metabolizer means the drug response is expected. Intermediate metabolizers mean their metabolism is slow, so higher drug level than expected. And then in the poor metabolizers, they have very slow or no metabolism, so higher level of drug and high risk of ADR. So an example of population ratios. So in this, the example is given of the means population ratios, that in a particular population what is the percentage, that how many of individuals could be having the ultra rapid metabolizers, how many could be extensive metabolizers, and how many are the intermediate, and how many are the poor metabolizers. So if we look at the population, then majority are the extensive metabolizers, then are the intermediate metabolizers, whereas ultra rapid and poor metabolizers are very low. So 1:5:3:1. Then an example of drug dosage, that how much drug should be given. So the ultra rapid metabolizers, because they are very rapidly metabolizing the drug, so drug is clearing very fast, so high dosage is required. Whereas to these individuals, means not high as ultra rapid, but slightly higher dosage is required. To intermediate, lower dose is required, and to poor metabolizers, very low dose is required. So an example of pro-drug dosage, this is the pro-drug dosage. So pro-drug means the drug which is helping this particular drug to effectively work. So this is the example, that how much dosage of this pro-drug is required. Okay, so this tells you that why the genetic variations are important to consider before designing the drugs or individual therapies or individual drug doses. Okay, now we'll talk about the precision versus the personalized medicine, that what is the difference. So personalized medicine is when we say that it is a broad concept, it is tailored to the individual. If we are only talking about the single individual, that how the drug is designed for that particular individual, that is known as personalized medicine. Whereas if we talk about the precision medicine, that means subgroup based approach, which is more practical. So NIH has given this statement. So they said that the precision medicine delivers the right treatment to the right patient at the right time. So that is the precision medicine. That means the right treatment to the right patients at the right time. That is known as precision medicine. It does not care about the single individual. It says that it should be the right treatment, and then it is the precision medicine. So we can say that the personalized medicine could also be precise. It could also be the precision medicine, because it might be given to that particular individual at right time at right dose. So that is the difference between the personalized medicine and the precision medicines. So personalized means that is there is unique customization. It is ideal, but it is difficult to achieve, because to each individual designing it is quite difficult. Precision, it stratifies the patients by biomarkers, genetics, and risk factors. So in that way, it is like for the subgroup we are designing this, and not to one single individual. So example is cancer therapy guided by mutation profile. So when we are designing the cancer therapy, what we will do, we will take all the patients, we will see their mutation profiles. So it is possible that in different we can convert those patients into groups. We can group those patients. So some group of patient might have single same kind of mutation, whereas other patients could have other mutations. So this is what is known as stratification. We can group those patients, all the patients, into different stratas or different categories or different groups, you can say, based on the biomarkers, their genetics, or the mutations. So this is the example that how the precision medicine works. So this does not take that much time as that of the personalized medicine, and both of them improve the efficacy and efficiency versus the traditional. So both improve the efficiency with comparison to the traditional, which was based on the one size fits all. So it is often used interchangeably in practice, but there is a subtle difference which exists. So you will see that the precision and personalized medicines, they're interchangeably used. So in place of personalized medicine, people might use the term precision medicine. So there is a very minute difference between both of them. Okay, so this shows you that the traditional medicine system, it believed that one size fits all the medicines. That means same medicine can work on all these patients. So what we do in precision medicine, we stratify them. We divide these patients on the basis of their disease subtype, their demographics, their clinical features, and their biomarkers. So in demographics, we means that which environment they are present based on their geographic characteristics. For example, if I have cancer patients, some are from the other continent or other country, so we will categorize them on the basis of their geographical distribution, that where they live. Those environmental factors are also considered. So this is how they are stratified, and then personalization is done. So patient individual, so we look at the preferences, clinical features, medication history, environment, their behaviors and habits, and biomarkers, and that is how the precision medicine is designed. Okay, so in this case also it is shown that how it works. So clinical, there's a clinical data, and then the tissue samples are collected, both are combined, and then personal omics data, in which we look at the genomics of that patient, transcriptomics, proteomics, metabolomics. So all the omics are studied, and then there is a library of biological samples. Then cloud computing, in which there is large scale data analysis is done, and then the personalized medicine is designed. So in this analysis, because we already have this information that which drugs, because when the studies are happening, experiments were taking place, we also see that how this, because at the group of patients the study is being conducted, which we call as the clinical trials. So during them we know that what could be the effect of this drug. So in this case, when we are talking about the large scale data analysis, what we are doing is we know the genomic characteristics of the person, so we are comparing it with the already available data, that how the different mutations can influence the effect of drug. So we compare with that particular data which is already available, and then we design the personalized medicine. So that means to design the personalized medicine, we require the data, means the pre-existing data, or we can call it as the information or the databases which are having that information, that how that particular mutation or how that particular variant can affect the drug response. If we do not have that data, then we need to experimentally perform these experiments, and then only we can design the personalized medicine. Okay, so this is all about the how the precision medicine is designed, and what is the subtle difference between the precision and personalized medicine. Okay, then in 2015, the president of US, Obama, he also took initiative towards this precision medicine. So he announced in 2015 the landmark US government program, and his vision was to shift from the average patient model to precision healthcare. So it was the major project of that time, and it was the All of Us research program. It collects genetic plus lifestyle plus environmental data from more than 1 million participants, and many countries have taken this initiative, in which now they collect the genomic data of each and every patient already available, and there is a biobank. So in biobank, the data is already stored for the research. So it encourages the use of genomics, big data, and AI in healthcare, and it also drives the development of targeted therapies and preventive care. It inspired similar initiative worldwide. For example, in UK, there is a 100K genome project, and now um, there are other genome projects also which are coming up. So it's very important, if we already have that data, then we can design drugs accordingly, because means now it has been realized, but previously when it was not realized, then people were using the drugs which were designed in foreign countries, and they were not that much effective in our country. So later on it was realized that different person have different genetic makeup, we have different genes, and we have different means you can say different isoforms of that particular gene which are expressed, different proteins which are expressed from that same gene. So we have different means response towards each of those drugs. So it's very important to study all these properties, all these genomic makeup of each individual before giving the drugs. So it is so what he mentioned is that it is they are developing the better approach to preventive care and medical treatments by helping the patients to gain the access to the health information so that they can collaborate in their own care, and it considering each individual's specifics like their genes, their environment, their lifestyle. So bringing new effective medical technologies to the market faster and building a research network of 1 million or more US volunteers. So this was the main objective or main initiative of this particular precision medicine initiative in US. And then human genome project and next generation sequencing, they also played important role to sequence the whole genome. So if we talk about 2003 when the human genome project was just completed, so that time the cost of sequencing that particular gene was around 3 billion. It was very much billions, then it reduced to millions in 2006, and then 2007 also it reduced from 20 to 2 millions. In 2008 it further reduced to 200,000 dollars, and in 2010 it reduced to $10,000. In 2014 it reduced to $1,000, and in 2017 it reduced to just $100. So this shows that with time and with the advancement of next generation sequencing techniques, how the cost of sequencing the genome reduced. So due to reduction in the cost, now we know that we can sequence more and more of genome. So that's why different projects got launched, and with the help of human genome project it was realized that there are total 20 to 25,000 of genes present in the human, and it was initially means its cost was in billions, and later on as we can see that reduced from billions to just hundreds. So it enabled the identification of disease genes and drug targets. So when we say drug target, we mean that drug target are the genes or the proteins which are responsible for the disease. So we design drugs to target those proteins. So they are known as drug targets. Means where the drug target, where the drug binds. So that are drug targets, and these are already present in the patient's body. Next generation sequencing leads to the faster, cheaper and high throughput, and then it allows the whole genome, exome and transcriptome sequencing. So not just DNA sequencing, but we can also sequence the exome, which includes just the coding elements of the DNA, which are exons, and transcriptome means we are also looking at the RNA. So transcriptome sequencing, generally it happens when we have the RNA, we convert it into cDNA and then we do the sequencing, that is referred to as the transcriptome sequencing, and it is the basis for the personalized risk prediction and therapy selection. Integration with artificial intelligence, that is AI, enables the predictive modeling. So we can also do this modeling with the help of AI, and it marked the beginning of genomics era in medicine. Okay, so this shows that how the human genome project has also played its important role towards the personalized therapy, because in personalized therapy we require the genetic makeup of the person. So previously it was very costly to do that. So even if we know the personalized medicine, but we cannot achieve it because it was very costly. But now, because of completion of human genome project and due to the reduction in the cost of sequencing, now this has become achievable. So we can means get the genomic data of any individual easily with the low cost. So engineering in precision medicine. So precision medicine, it depends not just on the genomics, but it also on the engineering innovations. So biomedical engineer design the tools to detect, monitor and deliver the therapies. It includes biosensors, implantable devices, biomaterials and nanotechnology. It enables personalized drug delivery and patient monitoring. And it also combines the biology, computer science and engineering for medical solutions. And one such example is of the insulin pumps, which are tailored to patients' glucose patterns. And then engineering of these things, it bridges the gap between the lab discoveries and clinical practice. So without engineering, personalized medicine would remain largely theoretical. So this shows that how important is engineering in the precision medicine. So these different things we'll see in the later slides, that means how important is the biomaterials and what are the biomaterials. So all these things are very important. Similar thing is mentioned here. The material chemistry, what kind of material is used in these biomaterials is very important. So polymer network to release the drug. So the biomaterials, because when we are designing the personalized drugs, but how to deliver them into the patient is also important. So we need some certain things or certain drivers through which we can insert the drug into the patient. So things with which we deliver it are known as the biomaterials, which enter into the body and which react with the biological molecules, so that are known as biomaterials. So those biomaterials could be those polymers which are wrapped, in which the drug is kept. So the polymer is the outside covering of the drug. When it enters into the body, the polymer degrades, it reacts with the body, and it get excreted out later on, but it releases the drug inside the body. So that comes under the material chemistry, in which we look at the material property, that whether it is safe, and means how much time it will take to melt and then release the drug, and means at which condition. Because for example, if I want to means want a drug to act in the stomach, or I want it to act in the other parts, so I'll design it in such a way that it does not get, means drug does not get released in your mouth, it get only released in a stomach. So I will design that in, means I will coat it around, means I will coat a biomaterial around that drug, which is which is only getting degraded at that particular pH. So these are known as the smart materials or smart biomaterials. So which respond to particular stimulus, and then they means perform some action. For example, they open up and release the drug. So similarly, these things come under the material chemistry, where we design these drugs by keeping those things in our mind. So they are the patient specific enzyme to release the drug cocktails. So combination of drugs can also be administered. Then comes fabrication. So there are certain materials, for example, if there is bone injury or some other body part is injured, so these biomaterials can also act as the, means the 3D biomaterials, which can act as a scaffold, which can support your that particular uh, broken region or broken bone. So they, this comes under fabrication. So these biomaterials can also act as the uh, scaffold in your body, or the means support, they can work in the supports. Then comes bioactive components, such as during the transplants, so allogenic transplants or autologous transplants or engineered cell transplants. So these are also the biomaterials only. Then patient data analysis. So the biopsy, for the biopsy also, the materials which are required to keep those samples in that particular material and then study them. In situ evaluation, so for this also the engineering techniques are required. Real time tracking, so these biomaterials can do the tracking at the real time. So all these things come under the engineering innovations, because with the help of these innovations, now we can design all these materials effectively. So one such example was of the insulin pumps. So this also we'll see in the later slides, that how they work. So now the applications of engineering in healthcare. So these are some of the examples, such as biosensors, which measure the glucose, cholesterol, and DNA mutations in the real time. So right now, you might have seen the smart watches, which measure your pulse rate. So they are the smart materials only. So means they come under the application of engineering, that how the engineering is helped to means assess all these properties in the real time. So they, and that come under the category of biosensors. And not just the smartwatches, there are some other equipments or the other machineries which exist, which could also measure your glucose, cholesterol, and DNA mutations at the real time. Then smart drug delivery systems, which release drugs only when it is needed. Then regenerative medicine, which is like stem cell engineering and tissue scaffolds. Then microfluidics, which is lab on a chip, which is used for the rapid genetic and biomarker testing. Organs on chip, which simulate the organ response for the drug testing. So these are some of the examples of the applications of engineering. Medical imaging advancements such as MRI, PET scans tailored for individuals, AI enabled devices which predict the disease risk and optimize the treatments, and engineering expands the precision medicine from theory towards the practice. Yeah, some of the examples are shown here, the MRI machines and different other machines which performs the CT scans, ultrasounds and PETs. Then the prosthetics and orthotics such as the prosthetic arm or prosthetic leg. So they are also under the example of this bioengineering, in which these are the, with the help of biomaterials, these are designed, and they are not just means means they are sensitive. So means right now there are different limbs or the robotic prosthetics which are coming up, so they are very sensitive. If you move it, then it also moves, so that is smart, that is like another part of your body only. So means, so right now with the help of this advancement in engineering, we can design the high level prosthetics which can mimic your same body part. Based on your movement, it will perform its action, and with the help of AI, it has now advanced way more. Then comes implantable devices such as the these are the cochlear implants which are used in the ear, and these are the pacemaker. So in the heart, the pacemaker is generally to regulate your blood flow and the heart rate. So these also come under this biomaterials. Then robotic surgery. So this surgery, all these things which are used, so they also come under advancement of engineering. And then wearable healthcares, which we have seen, the smart watches or the glucometers which check your amount of glucose in your blood. So glucose monitors, smart watches, fitness trackers, so they come under the biosensors. So this is how all these advancements have helped us to move one step more towards the personalized therapy. Then comes precision immunization. So means these are all like a smaller terms under a bigger term, which is the precision medicine. So precision immunization means when we are using the immune treatments or immune cells are involved, such as the vaccines or all those things, so they come under precision immunization. So immune system is highly variable among different individuals. So traditional vaccine, we all know that it was believed that same dose can act for all, but responses differ among all these individuals. So precision immunization, what it does is it uses the genetics and immune profiling for tailored vaccines. So it design the vaccines in such a way by keeping these factors in mind, the genetics and immune profile. For example, CAR T cell therapy, in which patients' own immune cells are engineered. So in previous session, we looked into the CAR T cell therapy. So in that case, we extract the cells from the same patient and we take it, and we at the surface or at the receptor of T cell, we change it with the chimeric antigen receptors, which are CARs. So this is how means we reduce the effect of the means host graft rejection. So we reduce that because we are using the same patient's um, cells, and then we are treating it and we are changing it, we are modifying it in such a way so that it can recognize the antigen, and then it can means the T cell can work well. So personalized cancer immunotherapy, which focus on the checkpoint inhibitors which are selected by the genetic biomarkers. Engineering enables the custom antigen design for vaccines, and it could reduce the adverse effects and increase the efficacy. So it represents next generation vaccines and immunotherapy. So this comes under precision immunization, and this figures shows the same thing, that how the precision vaccines are designed. So I'm sorry, actually I guess the light has gone. Okay, but recording will take place, so even if the class ends, but still there will be a recording, so you can see from there. Okay, so in the precision vaccine design, what happens is, if a natural, this is the example of the natural infection, and then later on the patient has shown the disease resistance, and if he is vaccinated, then what happens is the okay, so disease outcome could be fever, nausea, coughing, or it can also lead to death, organ damage, and hospitalization. So, in the precision vaccine design, what we do is we learn that how the infection affects the individual, whether it leads to disease resistance, and what happens after the vaccination. So, age specific human in vitro modeling is done, in which we look at the preclinical evaluations, in which we use the animal models to study the same things, study the effect of the drug, and then we use the perform the targeted clinical trials in which we perform those testing on the individuals, human beings, and then we have the system biology omics. So in this we capture the results, that what is the influence of the vaccine on different individuals, and we keep on modeling. So modeling means we see what dosage of that particular vaccine is performing what kind of action, and we also distinguish these individuals on the basis of their age, their gender, and then we see that how the vaccines work on these individuals. So this is how we build our data set. We built a database where we can store all this information, so which helps us to design the immunogen. So immunogen is a category of antigen only. So when we say antigen, that antigen is the foreign particle which enters in our body and to which our antibodies bind. So not all antigens are the ones to which the antibodies bind. They might not be immunogenic. So immunogenic antigens are the one which can generate the immune response inside the patient. So antigens are categorized into immunogens and antigens. So immunogens are the ones antigens which can bring about the immunogenic response in the patient's body. And then these are the adjuvants, which are the cytokines, small molecules. It also helps us to formulate the formulation development, that how to deliver these drugs, the hydrogels, cellular vehicles, viral vectors, microparticles, nanocarriers. So we keep our drugs in this. And then delivery routes, how to deliver that particular drug. So it could be transdermal patch, injection, or it could be through sprayable gels. So this is how the precision vaccine design generally takes place. Okay, then we'll talk about the stem cell engineering. So induced pluripotent stem cells, iPSCs, which are taken from patients' own cells, are reprogrammed. It can be engineered into any tissue types, because in the previous session we have seen that these stem cells are pluripotent in nature. Pluripotent means they can convert to any cell in the body, and it allows the disease modeling for personalized drug testing. It has potential to replace the damaged tissue and organs. So this comes under the regenerative medicine. Stem cell banks store the patient specific cells for therapy, and iPSC derived cardiomyocytes, they are used to test the cardiac drug safety. Genetic editing on these iPSCs, which are induced pluripotent stem cells, it allows correcting the mutations before the therapy, and it is central to future of precision regenerative medicine. Okay, so now let's see the healthy fibroblast and the patient fibroblasts. So in healthy fibroblast, the mutation introduction strategy. So this is a donor, also known as control. From him, we collect the somatic cells and we convert them into the fibroblast after reprogramming into SOX2. These are some of the proteins or the factors which are provided for the reprogramming of these fibroblasts into the control iPSCs, which are induced pluripotent stem cells. So when we say induced, that means these stem cells are induced. By the induction, they are converted into the pluripotent stem cells, and these were like the multipotent stem cells, and they are converted to the induced pluripotent stem cells. So after differentiation, they can convert into the cardiomyocytes, which are the heart cells. So genome engineering is done, in which the mutation is introduced. So after differentiation, they will convert into the cardiomyocytes with that particular mutation. So we can compare its phenotype with this. So this is the patient fibroblasts. And then mutation correction strategy. So patient with the known mutation, somatic cells are isolated, fibroblasts are obtained, then these factors are provided which reprogram these fibroblasts into the mutated iPSCs, which are induced pluripotent stem cells. After differentiation, it convert into the cardiomyocyte mutated with wild type factor. And in this case, we perform genome engineering, in which we correct the mutation and we differentiate into the cardiomyocyte. Both are the wild type traits are present. So in which we replace that mutated region with the correct copy of that particular gene. So in this we compare the phenotype to see the influence of mutation and with respect to the genetic background. So what mutation is present. So in this case we are introducing the mutation, and in this case we are correcting that mutation and we are comparing them. So this is how stem cell engineering is done, in which we are using the stem cells to study this, to study the influence. So it is mentioned that it is used to test the cardiac drug safety. Okay, so gene editing with the iPSCs. So induced pluripotent stem cells combined with the gene editing tools such as zinc finger nucleases, TALENs, and CRISPR. So they help in the genome editing. So how we can do? So examples are repairing the mutations which are causing the blood disorders such as sickle cell anemia, corrected iPSCs differentiated into the healthy cells for transplant, patient specific, it avoids the immune rejection, it can model the rare genetic diseases in vitro, and it is used to test the therapies before applying them to the real patients. It moves towards the curative therapies instead of just symptom management, and it is the major breakthrough in the personalized genetic medicine. So this is the comparison between the improved symptoms versus the sickle cell disease. So in sickle cell disease, the blood cells are in the sickle shape due to the change of one single amino acid in the that particular gene, which leads to the hemolysis and also vaso-occlusion in multiple organ systems. So what we do is we collect the autologous hematopoietic progenitor cells. So these HPCs, which are the hematopoietic progenitor cells, which give rise to the blood cells, so they are obtained. Ex vivo genetic modifications of these HPCs is done, in which we correct for this beta globin gene. In beta globin gene, there is a mutation which leads to this sickle structures of the RBC. So we correct that mutation and such that beta globin gene give rise to the healthy RBCs, and it leads to increase of the fetal hemoglobin. So CRISPR-Cas9 can help us to increase the fetal hemoglobin and addition of the variant hemoglobins by using the lentiviral expressing HbA T87Q. So this can be done. All these three different types of modifications can be done ex vivo for these hematopoietic progenitor cells. Then myeloablative conditioning is done, and reinfusing these genetically modified this hematopoietic progenitor cells are again engrafted back into the patient. So this improve the symptoms, reduce the hemolysis, and also reduce the vaso-occlusion. So this shows that how the gene editing of these iPSCs can help to cure the disease. Okay, so my internet is still not back. Okay, so engineering biomaterials. So when we say biomaterials, these are the engineered substances that interact with the body. They are used in implants, prosthetics, and tissue engineering, and they can be customized based on the patient's physiology. There are some smart biomaterials which release the drugs and respond to temperature and pH changes. There are biodegradable scaffolds which support the tissue growth, and then they dissolve after some time when that particular tissue is restored. Example is the stunts, which are coated with the drug eluting polymers. It reduces the risk of rejection, infection, and inflammation, and it enables the safer and more effective long-term therapies. So these are some of the examples. So this is the citrate polymer, which can be modified into multifunctional purposes, such as it can be made into the elastomer, fiber, porous, mycel, hydrogel, So they are multifunctional bioactive citrate based scaffolds which are used in various biomedical applications, such as they can act as the biovascular tissues, orthopedic tissue, muscle tissue, skin tissue, nerve spinal cord tissue, bioimaging, and drug gene delivery. So these scaffolds, these multifunctional bioactive scaffolds, can be used for all these purposes. So this come under the biomaterials, and with the engineering of these biomaterials we can advance these treatment therapies. So similar thing is mentioned here, that how they can act as the elastomer, bone graft, they can be nanofibrous, or they could be other grafts and vascular grafts, and then delivery vectors, bioimaging, they can also be helpful in bioimaging, tissue engineering, and delivery vectors. Okay, so in terms of drug delivery systems, the traditional drug delivery was the systemic and it also caused side effects. But with the help of precision medicine, let me okay, so with the help of precision medicine, it uses the targeted delivery for efficiency. Nanoparticles can carry the drugs directly to diseased cells. Example, liposomal doxorubicin in cancer, it reduces the toxicity. Closed loop systems such as insulin pumps release only as needed. Personalized dosing algorithms adapt to patient's physiology. It reduces the wastage and improves the safety. Drug delivery is central to making the therapy personalized, precise, and safe. So, drug delivery systems could either be liposomes, nanoparticles, dendrimers, hydrogels, carbon quantum dots, mucilage, polymeric materials. So, all of them can be used for the delivery of the drug, and we can also determine how much dosage is required. So, insulin pumps come under the another category in which these are like closed loop systems and they help us to achieve the personalized dosage. So these are uh, this is the pump with the tubing, and this is the example of the tubeless pump. So these have the biosensors which sense the glucose in the body, and they means with this they are the smart devices which decides, which know when to release the insulins. So these come under the smart biomaterials. And how it work is that insulin is injected into the fatty tissue below the skin. This is the pump, this is the insertion site, so this is the same. There is a tube which is shown here. This come under the infusion set. So how it works, this particular device, this is a smart device, this is a pump which knows that how much dosage is required at what time, and then what it does is it inject that particular insulin into the fatty tissue below the skin. Similarly, the example was of the liposomal doxorubicin. So this is a liposome, and then this is a drug which is the doxorubicin, which is used in the cancer. So functionalized liposome is there, and this loaded nanostructure is used for the drug delivery. Similarly, polymeric micelle can also be used for this drug delivery. Polymeric vesicle can be used. Then the nano emulsions, polymeric nanoparticles can be used, hydrogel nanoparticles can be used. So these are some of the new approaches in which the docs could be encapsulated and administered into the body. So these act as the drug delivery agents. This is the structure of doxorubicin, and this doxorubicin is hydrophobic in nature, and doxHCl is hydrophilic. So these are two types of dox and dox molecules. Okay, so now we'll talk about the smart polymers in therapy. So smart polymers are the materials that respond to the stimuli, so such as pH, temperature, and enzymes. So it is used for the controlled drug release at the target site. The example is the hydrogel that swells in the tumor microenvironment to release the drugs. So that's why they are known as smart polymers. That means they respond to the stimuli. So in this case, hydrogel swells in the tumor microenvironment to release the drug. They are biocompatible and biodegradable, and they can be engineered for timed or triggered release. They improve the compliance, less frequent dosing is required, and applications in cancer therapy, wound healing, and tissue engineering. It represents the future of responsive and patient specific medicine. Okay, so this shows the smart polymers which can respond to the stimuli such as light, temperature, magnetism, macromolecules, small molecules, and pH. So, on the basis of these conditions, they could show separation, they could show PTM proteomics, they could PTM is post translational modifications. So, these smart biomaterials can bring about those post translational modifications. They can be helpful in the tissue engineering, cross linker delivery, food safety, and sensing. Okay, so these are the smart polymers. Now we will see application of these smart polymers. So they can act in the scaffolds. So they support the stem cell growth in regenerative medicine as it is seen here. They can be used in the stunts where they release the anti-clotting agents in the cardiovascular patients. Wound dressings, so release antibiotics when the infection is detected. 3D bioprinting, smart polymers used in the custom tissue and organs, such as in this case artificial medicine. So this is after the 3D bioprinting of the smart polymers. They can act as the artificial muscle. Responsive gels, adjust the stiffness for the bone cartilage regeneration. An example is temperature sensitive gels for burn healing. And they are adaptable to each patient's healing process, and they expands the scope of engineering driven precision healthcare. So this shows the application of smart polymers, that in different fields how they can be used. They can be used in the surgical suture, correction devices, artificial muscle, cell scaffold, microinvasive surgery. Okay, so another term is theranostics. Theranostics means the combination of therapy and diagnostics. That is theranostics. So theranostics is combining the diagnosis plus treatment in one platform. The examples are nanoparticles that detect cancer cells and then they release the drugs. So ET tracers can both diagnose tumors and deliver the radiotherapy. They are personalized because patients get the real-time monitored therapy, and it enables the continuous feedback during the treatment. It reduces trial and error drug use and growing rapidly in oncology, neurology, and cardiology through integration of precision engineering and imaging in this case. So this is the theranostics in which we combine the diagnostics plus therapy. So targeted therapy in which we take the information of nucleic acids, proteins, anti-cancer molecules and peptides. All this information is available. And imaging and diagnosis, so in which we have the MRI, radionuclide imaging, computer tomography, X-ray, ultrasounds, and then we also have the nano agents, radio-labeled isotopes, plasmonic nanobubbles, liposome, polymeric micelle, and quantum dots. So all these fields are combined and then they give rise to theranostics, in which we combine all of them in the one platform and patient can get the real-time monitored therapy. So now we'll talk about the smart devices which are there in healthcare. So such as wearable track vitals, which track the heart rates, sleep, blood, glucose and oxygen, continuous monitoring, early detection of abnormalities. So if you're wearing all these gadgets, then you will know quite early that whenever some abnormality happens which is otherwise not recognized. So AI analysis patterns which alert the patients and doctors. So with the help of artificial intelligence, it will track the pattern. It will see that what is the normal pattern, and if it is abnormal then it will directly give you the alert. So it will not just help the patients but will also alert the doctors. Smart watches detect the atrial fibrillation, sleep apnea, etc. So data personalized for each patient, because in this case the smart device which you are wearing, it is personally for designed for you. So it will give your vitals. So in that way it is personalized for each patient. It supports preventive care and lifestyle adjustments. Devices can be synced with EHRs, which is the electronic health records. So that particular thing will also be recorded in some data records, so that you can come back and check those records, that how the different vitals are varying with time. It forms the backbone of digital precision medicine. So this comes under the digitally storing all that information. So similarly it is mentioned that those smart devices, smart fabric, smart camera, fiber optic, XYZ, so the users, they all work as the data acquisition devices in which they are acquiring the data of the user. Then with the help of Wi-Fi or the Bluetooth or other devices, this information is now concentrated, it's aggregated on the it is aggregated, it is adapted, pre-processing could be done, and feature could be extracted. So this comes under the data concentration, in which you are concentrating the information which is coming. Then we will process that particular information. And then the participants, so this is also conversible. So the pharmacy, research, insurance, emergency unit, physicians, so they are the participants here, which will use that particular information, and then they will share this information. Real-time alerts can be given, data could be analyzed, it could be stored, and it could be visualized. So this come under data processing. So this shows that how the smart devices can help you maintain the data digitally, and it will also give you the alerts real time, and you can also share it with your doctor. So it is like the personalized information of that particular patient. Then another term is organs on chip. So this is there are microfluidic chips which mimic the human organs. For example, lung on chip, as you can see, heart on chip, liver on chip. So these are some of the examples which allows the drug testing without the animal trials. So these microfluidic chips, they resemble your own organs. So instead of using animal models, we can study the effect of drugs on these chips. So in that way, these are the personalized chips which can be made from the patient cells. So the cells can be obtained from the patient, and then we can see how the drug response is observed on these cells. So that is how we can we can simulate the personalized drug reactivity in that particular patient. So it predicts the drug toxicity before giving it to the patient, and it is faster, it is ethical, it is more precise than the traditional models, and it can also model the rare and complex disease in the lab. It has potential to transform the preclinical personalized drug testing. So as you can see, because we do not require animal models any long, and because these cells are obtained from this patient, so we will be very sure that what will be the influence of the drug. We will be able to see the toxic effects or the positive effects of the drug. So that is how this organ on chip can help us to design this personalized drug testing and making it more relevant or more potential. Okay, so now if we are having all this information, then there are some challenges also. So big data and artificial intelligence challenges in this precision medicine. So we all know that this precision medicine is generating the massive data, because we are taking the information at the level of genome, proteome, and clinical. So there is a lot of information which is present. So challenge is to integrate the diverse data sources. Now we have all these data, but how to integrate them, where to keep it, how to integrate, that is also one big challenge. We need the secure storing and sharing and analysis. We cannot make the data of each and everyone open to everyone. We need to provide some level of security. We need to provide some place to store that data, and we also need to analyze that. So that are also the another challenge. There is the risk of data privacy breaches. So if that data is used by some other agency or other individual to harm them, then also it is very bad. So there is a risk of the breaches of the safe privacy. So AI ML models, they help to identify the patterns from the data, because we have big data, we have lot of data, and to analyze it for an individual is very difficult task. So we can take the help of artificial intelligence and machine learning to model that, to analyze those patterns from the data and predict the outcome of the patient, drug response, and disease risk based on the already available information. It can model it. Ethical concerns are the bias, misuse of genetic data, and big data management is critical for the future of precision medicine. So this is very important, because if we have all such data, then we also require the proper management systems and proper analysis of all those things. At the same time, also protecting the individual data, and means it should be private, it should not be accessible by everyone. So these, if you're interested, you can go through this particular debate which was published in the BMC Medical Informatics and Decision Making, which talks about the big data hurdles in precision medicine and precision public health. Okay, so now we'll talk about the genome editing in personalized medicine, which we were previously discussing. So, genome editing means when we are editing the genome, we are modifying some components of the DNA, some DNA sequences. And the tools which we can use are the ZFNs, which are zinc finger nucleases, TELENs, CRISPR-Cas systems, which allows the correction of disease-causing mutations. Key role in tailoring the therapies to individual genetic profiles. It can inactivate the harmful genes or insert the beneficial ones. It has the potential to cure rather than just manage the genetic disease. And it revolutionizes the personalized medicine by enabling the patient specific genetic correction. And it is ethical and safety considerations remain critical. So, for example, in this case, this is a cancer cell, liquid biopsy samples in which the tissues are collected from this particular cell. Circulating cells free DNA and cancer stem cells could be used as the biomarkers. Next generation sequencing can be done, which help us to identify the mutated gene in these cells. And then we can use the CRISPR-Cas9 to study those mutations, and means we can either break those genes and use the donor DNA to cure that mutation, or we can just um, means convert that gene to a defective gene so that it does not express any longer. So this comes under the personalized oncology, and this shows the potential of genetic or genome editing in this field of personalized medicine. Okay, so now we'll quickly go through all these techniques. So in the previous sessions, in previous week sessions also, we have individually gone through zinc finger nucleases, TELENs, and CRISPR-Cas9. So again, once again, we'll give a means we'll go briefly into all of them. So zinc finger nucleases, these were the one of the earliest genome editing tools, and we have seen that it fuses the DNA binding zinc finger proteins with the nuclease domain. So zinc finger nucleases are the nucleases which cleave the DNA. So we add the DNA binding domain to this nuclease domain, and that is how the zinc finger nucleases are designed. They targeted means they perform the targeted cutting at specific DNA sites, because the DNA binding zinc finger proteins which are bind to these nucleases domains, these zinc finger are very specific to the particular DNA sites. And we have also seen that when we design those zinc finger proteins, then that particular category bind to particular triplet of the nucleotides, so that is very specific binding takes place. So in that way we can design zinc finger nucleases on our zinc finger proteins in such a way, means if we know where we want to target, so we can design these zinc finger protein units ourselves, and we can modularly construct this zinc finger nuclease ourselves. So example is targeting of BCR-ABL in chronic myeloid leukemia, and it is used in the studies to overcome imatinib resistance. And the clinical application is also in CCR5 disruption in HIV research. So we have seen that this CCR5, this is the receptor at the cell surface to which the HIV binds, and then it releases its genome, and that is how its infection starts. So if we have these systems, REDF and TELENs, CRISPR-Cas9, then we can edit this particular gene in such a way that it no longer give rise to this protein. So if this protein is not synthesized, there will be no receptor on the surface of cell. If no receptor on surface of cell, so HIV won't be able to bind or integrate its genome. So in that way, with the help of these genome editing strategies, we can lead to the disruption of certain genes which can cure the disease. So they are high specificity but technically complex to design, and in that way pave the way for the newer and simpler tools like CRISPR. Okay, now we'll talk about TELENs. So these are the transcription activator like effector nucleases. It is easier to design as compared to ZFNs, and it recognizes DNA sequences with modular repeat domains. It is used in the cancer therapy, and one such example is of the HPV, human papilloma virus E6E7 oncogene targeting. It is applied in CCR5 gene editing for HIV resistance, and it shows promise in treating the genetic and infectious disease. It is still less popular as compared to CRISPR due to its efficiency. But when we were talking about TELENs, then we have seen that these are effective if you want to treat something in the mitochondria for which the CRISPR was not effective. And it is the important stepping stone in the evolution of gene editing tools. So this is the paper in which they have discussed about the how the TALEN-mediated targeting of HPV oncogene ameliorates the HPV-related cervical malignancy. So in this they have talked about the HPV oncogene E7, that how its um, activity reduction can be brought about by this talens, in which there is fork1 nuclease, and then this is the tail, so which is the transcription activator like effector, which binds to the specific site in the DNA. And then after the knocking down of this, how it brings about its effect in the human papilloma virus related malignancies. So it reduces the malignancies. Okay, now we'll talk about CRISPR-Cas9, which was the third approach. So CRISPR stands for clustered regularly interspaced short palindromic repeats, and the Cas9 is the another protein which cuts the DNA at the targeted sites, which is directed by the guide RNA. It is simple, cheap and more efficient as compared to ZFN and talens, and it has broad applications in diagnostics, gene knockouts and gene corrections, and it is also used in the CAR T therapies, in which we change the receptors at the surface of T cells. We engineer the immune cells against cancer, and one such example is of PD-1 knockout in T cells, which enhances the cancer immunity. It is applied in the hematology. Hematology means where we are studying the blood cells such as sickle cell anemia, beta thalassemia trials, and it is the leading tool for the personalized gene therapy research. So this is the example of the patient from where we collect the somatic cells as well as HSCs, and then we reprogram them to the iPSCs, so which are induced pluripotent stem cells, and these are hematopoietic stem cells which give rise to blood cells. We combine them and we use CRISPR-Cas9 to correct these hematopoietic stem cells. These are the corrected hematopoietic stem cells and these are the corrected iPSCs. Their differentiation is done into the hepatocyte-like cells. They are endothelial cells which are transplanted into the mice for analysis. And in this case, these corrected HSCs are further inserted back into the patient. So this is how the gene editing can be used to treat the disease state by bringing about by either adding the new gene, or by the disrupting the faulty gene, or by correcting the some gene. So this is the example of CRISPR-Cas9 in the therapy, that how it is used. And then comes CRISPR screens. So these are like genome-wide screening, which identifies the essential genes in the disease. So because whenever a person is suffering from any disease, so first of all it's very important to identify that which gene is responsible for the disease, because multiple genes could be responsible. So in this screens we identify those essential genes in the disease. There could be different studies which could be performed to find out these essential genes. So one such thing could be subpool screening, which focuses on the pathway of interest. Then patient derived organoids, which are used to test the therapy in vitro, or patient derived xenografts, which model the therapy in vivo, and it helps to map the genetic vulnerability in cancer and other diseases. It also enables the precision targeting of tumor specific pathways. It reduces the trial and errors in the therapy development with the help of these screens, and it is the foundation for the large scale precision oncology approaches. So there is another paper which was published in Nature Reviews. So if you're interested, you can go through this, in which they have mentioned that the difference between the pooled CRISPR screens versus the arid CRISPR screens, that how by using these screens we can identify the essential genes which are responsible for the survival, and which genes should be considered to perform the drug treatments and resistance. So all these things can be done. So if you're interested, you can go through this. Okay, then comes CRISPR UMI, which is the unique molecular identifiers. So it combined the CRISPR editing with the barcoding technology. So it tracks cell to cell heterogeneity in response to gene editing, and it allows the study of clonal dynamics after editing. It reveals how different subpopulations respond to therapies. So there could be subpopulation of DNA, so we can label them with the help of these unique molecular identifiers, so that later on we can group them and we can study them, that how the drugs behave, how they respond to therapies, each individual group. So this is important for understanding the cancer resistance mechanisms, and it improves the reproducibility and accuracy of CRISPR studies. It helps to refine the patient-specific gene editing therapies, and it advances the precision monitoring of the genome editing outcomes. So this is how different UMIs, which are unique molecular identifiers, can be used, and we will be able to know that what is the influence of the, means how different clones vary and how they behave to the different drugs. So this is the CRISPR UMI positive selection, in which this shows the high penetrance and low expansion, and this is the low penetrance and strong expansion group. So measure of the incidence rate and the effect size. So with this, we can uniquely label the individual DNA molecules, and we can group them and study them. That is the main purpose of this CRISPR UMI. By barcoding them, we can track the heterogeneity in response to the gene editing. Okay, then there are some clinical trial case studies for the CRISPR edited T cells. So first in human clinical trial took place in 2016 in China. So the patients advanced non-small cancer cell lung cancer, advanced non-small cell lung cancer, so NSCLC, and approach was PD-1 gene knockout in the T cells using the CRISPR. So they edited the T cells, reinfused them into the patients, and the goal was to enhance the immune response against the cancer cells. It showed safety and feasibility, though modest efficacy, and it proved that CRISPR editing is clinically possible, and it opened the path for many ongoing trials worldwide. So in this case, the patients, we have taken the cells, normal T cells from that patient, and used the CRISPR-Cas9 to edit it, and then we have um, brought about the PD-1 gene knockout using the CRISPR technique, and then endogenous TCR knockout, and then we have used the lentiviral delivery of the CRISPR edited T cell into the body, and then it was injected back into the cancer patients. So its goal was just to enhance the immune response against the cancer cells. So this is the clinical trial case study in which it was observed that CRISPR editing is clinically possible. We can clinically perform the knockout of particular genes using the CRISPR technique, and we can also use the we can also insert that CRISPR edited T cell with the help of lentiviral vector delivery system, and which we can use to insert back into the cancer patients. Okay, then comes gene editing in the cardiovascular diseases. So targeting PC3 mutation in the hypertrophic cardiomyopathy, which is the example of the CRISPR correction in human embryos. PRKAG2 syndrome editing used to restore the cardiac function in the models. Gene editing could prevent the sudden cardiac deaths in the genetic heart diseases. It has the potential to repair the mutations at the early stages. So that side comes under preventive therapy. That in the early stages only we are curing it. So it is like a preventive therapy. It's preventing the disease state. Still experimental but highly promising. And it highlights the role of personalized genome editing beyond the cancer. It could reduce the global burden of inherited CVDs, which are the cardiovascular diseases. So these are some of the figures which shows that means how we can do it. So these are the iPSC cells. First of all, the patient from where we have obtained the primary cells, they could be reprogrammed with the help of reprogramming factors. They could be converted into the induced pluripotent stem cells, and in these pluripotent stem cells we can obtain their DNA, and we can correct the genes with the help of CRISPR-Cas9. So then we have the edited isogenic iPSCs. Because we have performed the editing at the level of genes, we can differentiate them, and differentiated iPSCs can further be transplanted into the patient. So this is how, with the help of CRISPR-Cas9, we can perform the gene editing, and means we can perform it at the early stages also to reduce the cardiovascular disease symptoms. Okay, so in this case it is mentioned what kind of delivery systems could be used. So viral vectors, it includes the adeno-associated viruses, lentiviruses, whereas in the case of non-viral delivery agents we have the liposomes or the polyplex or lipoplex. So these are the delivery systems which deliver the CRISPR-Cas9 into the circulation of the specific organs. Okay, now gene editing in the infectious diseases. So infectious disease like HIV, so disruption of CCR5 receptor to block the viral entry, so which we have seen in the case of ZFN talents, that how they can be used to knock down the CCR5 gene, which will no longer give rise to the surface receptor to which the HIV binds. So it was inspired by the Berlin patient CCR5 mutation, which leads to HIV resistance. Then comes HPV, human papilloma virus. So CRISPR targeting of the E6 and E7 oncogenes inhibit the cancerous transformation. HVV, CRISPR used to target the viral DNA in hepatocytes and potential to clear the chronic viral infections. It represents the new frontiers of antiviral therapy, and it offers hope for the disease previously considered incurable. And trials are still ongoing to test the safety in the human patients. So this talks about the infectious disease by these viruses and how we can use them, ZFN, talent, and CRISPR-Cas, and they bring about double-stranded break. Later on, what kind of repair mechanisms takes place, the outcome could be variable. So there are different clinical trials which are ongoing. So early trials were the ZFNs in HIV patients, in which the CCR5 editing was performed. Then Telin was also tested in leukemia and HPV related cancers. CRISPR dominates most clinical trials, more than 50 ongoing globally, and diseases targeted were the blood disorders, cancers, viral infections, and cardiovascular diseases. And most trials are still in the phase 1 or phase 2. As you can see, majorly are in the phase 1, then some have entered into phase 2, some have entered into phase 3, and very few are in the phase 4, which are the approval phase. So encouraging results, but long-term safety must be proven. And regulatory challenges around the germline editing, because in the case of germ cells, the editing still have the regulatory challenge due to ethical concerns, and many other reasons exist. So clinical trials shows the rapid translation from lab to bedside. So in conclusion, personalized medicine integrates the genomics, engineering, and artificial intelligence. We have also looked into the historical roots. Ayurveda and Archibald Garrod, they have shown that long-standing interest in the individual treatments. Engineering innovations make the precision therapy feasible. Genome editing tools such as CRISPR, TEL, ZFNs, they are transforming the therapy. And currently we are focusing on the cancers, genetic disorders, infectious diseases, and future keeps the broader use in the preventive medicine and regenerative therapies. The challenges right now are the ethics, safety, data privacy, accessibility, and the long-term vision is that the medicine where the every patient receives the right treatment at the right time, which is the precision medicine. So this is how the medicine concept has evolved through time, traditional medicine to evidence-based medicine to the precision medicine, in which there is stratified medicine, personalized medicine, and individualized medicine. And these are the benefits of artificial intelligence in healthcare. So it has enhanced the diagnostics and imaging, personalized treatment plans, predictive analytics for patient outcomes, and efficient healthcare operations. So this is all about today's session. Now we'll move to the practice MCQs. So the first question is, what does personalized medicine combine to create the tailored healthcare? So option A is environmental and genetic characteristics. Option B, genetic and phenotypic information. Option C is only genetic characteristics. And option D is genetic, phenotypic, and environmental characteristics. So you need to mention that what does this personalized medicine combine? Okay, so the right answer is option D, because we have discussed that in personalized medicine we look into the genetic, phenotypic, and environmental characteristics. All of them are important. Okay, so next question is, what limitation does personalized medicine overcome? Option A, lack of genetic information. Option B, one size fits all therapy. Option C, lack of environmental data. And option D is cost of therapy. So you need to tell what limitation it overcomes. Okay, so the right answer is option B. So traditional medicine believed that one size fits all. But now with the help of personalized medicine, we know that different individuals behave differently to different drugs. So that particular limitation has been overcome with the help of personalized medicine. Okay, so next question is, what are the three primary humors in Ayurveda? So mainly they were doshas, but if it is mentioned humor, so it is also one of the same thing only. Option A, Vat, Pith and Cough. Option B, air, water and fire. Option C, blood, phlegm and bile. And option D is earth, space and light. So this is quite straightforward. So right answer is option A, which is Vat, Pith and Kapha. These are the three doshas. Vat includes ether and air. Pith includes fire and water element, and Kapha includes earth and water element. These are the seasons which are shown. So Vat is connected to the fall or autumn. Pith is connected to the summers, and Kapha is connected to the spring. Okay, next question is, what term is used interchangeably with personalized medicine? Option A, evidence-based medicine. Option B, standard medicine. Option C, precision medicine. And option D is alternative medicine. Yeah, so this is also well discussed in today's session. So the term is precision medicine. So the term personalized medicine is often used interchangeably with the precision medicine, and both refers to the medical approaches that tailor the treatment and prevention strategies to an individual's genetic makeup, its environment and lifestyle. And it aims to move away from the one size fits all treatment towards the targeted and individualized therapies. And another definition for the precision medicine is that right treatment given to the right individuals at the right time. Okay, so next question is, what is the purpose of smart polymers in the precision medicine? Option A, to increase the weight loss. Option B, to change the properties in response to the specific stimuli. Option C, to enhance the color in the textiles. And option D is to harden when exposed to light. So you need to tell the purpose of these smart polymers. Okay, so this is also straightforward. When we were talking about the biomaterials and smart polymers, then we discussed that why these are known as smart polymers, because they change their properties in response to the specific stimuli such as light, temperature, magnetism, macromolecules, smart molecules and pH. So these are different stimulus to which these smart polymers react to, and they change their properties. Okay, so next question is, what is the role of stem cell engineering in precision medicine? Option A, to increase the muscle mass. Option B, to repair the damaged tissues and model diseases. Option C, to enhance the skin appearance. And option D is to create the new organ systems. Okay, so this is also straightforward, to repair the damaged tissues and model diseases. So for that particular reason, we obtain the stem cells. Okay, so next question is, what is the function of separators in precision biomaterials? Option A, to detect analytes. Option B, to release the payload. Option C, to attach to the molecules or cells for separation. And option D is to degrade the materials. Okay, so let's see. So right answer is option C, that is to attach to molecules or cells for separation. So this figures shows that how this separator is separating these from this mixture into two individual components or two individual cell types. So example is disseminating cells, and this is a scaffold. It is separating the immune cells from the metastatic cells. So that is the work of the separator in the precision biomaterials. So in precision biomaterials, separators are designed to bind or attach to specific molecules, cells or particles so that these targets can be isolated or separated from a mixture. They often use specific ligands, antibodies or surface chemistries to achieve the selective binding. This enables purification, diagnostic analysis and targeted delivery processes. So separators function mainly in selective attachment and isolations, and they do not have any role in detection, payload release or degradation. Okay, so this is the another excerpt which I obtained from this particular ResearchGate article. So if you're interested, you can go through this, that other than separators, there are some sensors, responders, controllers and processors also, which are there in the biomaterial design. So these are the operations. These are different units and these are the work of these each individual unit. Okay, next question is, what does the term theranostics refers to? Option A, diagnosis only. Option B, combination of diagnostics and therapeutics. Option C, drug delivery only. And option D is monitoring the treatment response. Okay, so this is also straightforward. As the name suggests, theranostics, that means it is combination of therapy and diagnostics. So this is theranostics. Next question is, what are organoids produced from? Option A, blood samples, option B, tissue cultures, option C, stem cells, and option D is biopsies from the cancers. So you need to tell the organoids produced from. Okay, so let's see. So the right answer is option C, that is stem cells. So organoids, these are the miniature simplified versions of the organs which are grown in vitro that may make the key structural and functional features of the real organs, and they are produced from the stem cells, either from the pluripotent stem cells like embryonic or induced pluripotent stem cells, or from the adult stem cells from specific tissues. And these cells are cultured under special 3D growth conditions that allow them to self-organize and differentiate into multiple cell types, forming the structures resembling the real organs. Yeah, so this is how the organoid culture behaves. This is the ECM, this is the 3D culture in which it is placed, and this is how the samples are obtained. So they could either be obtained from the embryo or the human tissues, iPSCs, tumor cells, healthy tissues, or ESCs. So floating spheroid, cancer cells, somatic cells. So this is how the organoid culture behaves and it is produced. So next question is, what is a trade-off in personalized medicine? Option A, convenience and cost. Option B, convenience and privacy. Option C, effectiveness and regulation, and option D is speed and accuracy. Okay, so the right answer is convenience and cost. So this is the trade-off in the personalized medicine. We have to trade off for the convenience and cost. Okay, so this is all about today's session, and I hope you have understood everything which was covered in today's session. So if you have any doubts, you can ask in the next session, and then we'll also be having the additional session on the 16th October. So if you have any doubts and if you want any clearance on any topic, then you can let me know in the comment section. So we'll cover all those topics on the 16th October, and in that session I'll be keeping all those MCQs and assignment questions. I'll be keeping the previous assignment questions as well as the current assignment questions, so that you get prepared, you get to revise all the things which you have covered in all these 12 sessions, and so that your exam goes well. So that's all for today's session, and now I'll be ending the session. So thank you very much and see you in the next session.